Hello everybody, thank you for joining us. I'm very pleased to have the opportunity today to discuss about the insights of adding analytical traits into a breeding pipeline. So don't hesitate to use the chat to ask questions. I will take a few minutes if we have some at the end of my speech to answer. We wish to take some time today to discuss this subject as at Dorian, working for 30 years globally in IT for research in life science. We are confronted with multiple breeding programs with their own goals and processes. And some are particularly focused on developing food product crops with the objective of improving overall quality, chemical compositions, or nutritional value. And apples, blueberries, wine grapes, tomatoes, peanuts are a few examples of food products that were improved in the past decades through plant breeding. As any crop, the food product-oriented crops are bred with the objectives of increasing agronomic performances, for sure, to sustain the crop productivity. But plant breeding, as it's at the crossroads of multiple activities, need to consider all the aspects that are relevant for both the growers, like disease resistances, for example, but also the consumer consideration, like nutritional properties or flavor attributes. So in order to control all these aspects, the breeding departments need to involve multiple skills who collaborate and tools and methods that will be interconnected. Today, I will take some time to share our experience on what we have to think about in terms of data and process management at introducing analytical information into your breeding pipeline. I will first work through the insights and the organization to put in place to optimize the management of these new observations. Then I will share some experiences on some solutions which can be settled to optimize the management of your samples and share efficiently the results. And I will conclude by introducing some tools which support breeders into the decision making to confront all this new information with phenotype or genomic. Depending on the food product and the market targeted, the type of traits to track can be very diversified. So what information the analytical process generates and how to characterize it. So we can speak about the chemical traits, for example, the increasing of amount of micronutrients or bioactive compounds which modify fat and oil compositions or improving carbohydrate quality or protein quality. We can speak about processing traits, for example, for the wheat and the measurement of its capacity of bread making, or for vegetable with their capacities of conservation or cooking. The flavors, taste, for example, the sweetness of fruits or berries, and any consumer-based sensory characteristics. So in this context, the plant breeding need cooperate with other fields to characterize the traits targeted and understand the biological processes implied. And the resulting scientific methodology will particularly lean on the quality of observation measures. And the scientists will need to ensure the quality of the data at the source. So as we see here an example, defining traits dictionaries and standard operating procedures to capture them. This is even more important when the observations are captured manually by less educated staff. Then it is important for the scientists to guarantee the confidentiality of the data, as the information collected is by essence produced by the R&D. The processes need to be protected and the access controlled. So Dorian, as you see a few screenshots here, with its digital tools, structure the traits management with unified and standardized dictionaries and unified forms to guarantee the confidentiality and security of data entered. In many cases, the characterizing analytical traits results from complex workflows to put in place to recover them. So what are the challenges by setting up new information source in the breeding pipeline? R&D department face with new challenges. So first, there can be some traits which need to be measured at a specific vegetative development. So for example, for the very specific use case of vegetable soybean, the measurement of an anti-nutrients concentration is optimal as the stage R6 to characterize the sucrose content of the seeds at maturity. So in this case, the R&D department should set up an organization to track the vegetative developments and some digital tools to rescue by proposing warning when the tentative stage is reached. Then, what technical solutions to measure it? To measure efficiently the targeted traits or its correlated observations. For example, we think about cocoa beans in order to make flavor tests of chocolate, the processing of cocoa beans. The R&D organization need to set up a cocoa processing laboratory, which requires investments and machines, and the R&D department need, with this installation, to set up some quality procedures with device calibration or maintenance plans, and it is the guarantee of the quality of the data. Last but not least, to manage other activities, the R&D departments need to invest on resources to track this process. And we can easily think about the machines, as discussed just before, or the human resources, which need to be trained to track and manage these processes, but also some data management system to store this information. So to conclude on this first part, I would say that IT solutions should address most of the challenges of integrating analytical processes into a breeding pipeline, and I will give a special highlight on this in the next part. I would like to discuss about now sampling challenges and what data management solutions we can set up for analytical work. Analytical traits come from samples which are collected on cultivars planted in field or warehouses. And sampling in this context is very important, particularly if the analysis of the analytical traits are costly or time-consuming. So the breeders often make a pre-selection of cultivars that will be collected and analyzed. And on the other hand, if you run specific sensory analysis, the harvesting of samples need to be voluminous to guarantee the most of the viability of results. As the cultivars to analyze are selected in the field, there should be an interconnectivity between field and labs to optimize the workflow, to collect, order, or label the samples, and finally also share results. And standard operating procedures should be described to guarantee then the quality of the data captured by the lab. Another point which can be important in this area, if anonymity is required to guarantee the reliability of the tests, which can be very important in GLP certifications. Some particular attention need to be apported in the identification or in the tracking of the samples along the process. So thanks to our experience in laboratories integration into a breeding workflow, we now propose some database structure and some processing tools to set up a collaborative operating procedures. And this diagram you can see here is illustrating some software architecture which shows how within the same unified system, specific breeder-oriented structures are directly connected with specific design structures for labs. And sharing same processes, the field and activities and the laboratories benefit from a common tool addressing their shared insights. I would like to illustrate this by one particular example of process optimization settled in cooperation by field and lab activities. It is the delivery of a specific sample collection form to optimize the laboratory processing on multiple analyses of cultivars. If the list of analyses vary on all the cultivars, it can be time-consuming for the labs to organize the samples in plates per profile or analysis. So the project stakeholders sit together to propose an automated sample collection form for the field workers, helping them to organize the collection of the samples per analysis profile to help then lab managers. The samples arriving in the lab in the optimized order was saving a lot of time into plate preparation, cherry picking. And the field staff accepted to reorganize the order of the sample collection because the identification in the field was correctly defined into their data management system. And within the software solution provided by Dorian, the breeders had a map to help them to order the samples, and then they can print this map, or they can synchronize also on tablets. The uniformity of the systems between lab and field enable to guarantee the integrity of IDs and results. And people in charge of lab analysis with this organization saved a precious time and of course reduced their risk of mistakes running their analysis. Let's discuss about data management. A collaborative data management should offer further benefits to integrate analytical information. First, in terms of data collection and data centralization, I would highlight the capability to build a specific threshold for the storing of voluminous data, as these activities can generate a lot of data. In terms of data collection, the capacity of the databases software to communicate directly with measurement devices or with mobile application dedicated to capture observation. We can think about robots or sequencers and so on. Also, as we discussed in the example before, the use of a collaborative database system should enable to share in real time the information without any double copy of information, just by dedicated user panels in the software to view the correctly addressed information. And comprehensive data management system offer the possibility to guarantee the compliance with the business processes, which are particularly documented and implying multiple stakeholders. So we can see some examples of to-do lists to do within a software to follow a process. And these systems can be also highly recommended when some certifications are targeted. It proposes, for example, some solutions to track KPI, which are very crucial for quality levels. And I would like to illustrate this part with one concrete example of software architecture we deployed. We set up a centralized data collection system for sensory analysis lab to capture evaluation from a consumer panel in real time. With the application Index Mobile, we configured multiple users forms with the descriptors for each observations. And this documentation was very important for them to run descriptive tests in a large panel of consumers. This installation with a tablet or PC for each participant ensured also the anonymity of the observations, and it was synchronized with a centralized database, enabling to share in real time the results. And with the deployment, the people involved in this process saved a lot of time in first recovering the results, which was directly synchronized, and into also data validation, which was very important in this case, launching analytic and hedonic tests. Let's discuss about now data valorization. It opens the last point opens the discussion on how to integrate such analytical information in the breeding decision-making system. And Dorian, providing a data management system which supports analytical processes integrated into a breeding activity, proposed also some solutions to visualize such information and to confront with phenotype or genetic information. First, as you can see here, such activities deal with complex biological models which imply various factors and that generate a lot of information. So it is important to characterize the variability of cultivars' responses, and multivariance analysis can respond to this insight. And Dorian offers some user-intuitive tools to visualize and analyze these results. We propose also reporting tools, which enables also to characterize through graphical patterns the cultivar profile and to communicate on its sensory or nutritional qualities. Another example, this analytical information should be analyzed to classify also the cultivars and build new breeding clusters. So Dorian proposed to integrate with data management system algorithm of hierarchical classification, and they can be used on numerical or qualitative data. And such tools are also used in order to build new breeding index, which can combine phenotype, genetic, and analytical information. And my last example is a concrete example of the use of tailored breeding index resulting from descriptive results and genetic information within a complete workflow of marker assistance selection. The breeder with its specific panel transmits a list of cultivars to analyze to a sequencing lab. This list results from observations and pre-selection in the field. It selects a list of analyses to do, which are in real time transmitted to the lab manager. He receives this list and he can plan its analysis with its cultivars samples listing and the request of the breeders. They collect the results, and once collected, stored in the database, after a process of data validation, the lab can calculate an index. So, example of data which are imported into the database, visualized in real time, and the lab can generate new breeding index, which are in real time visualized by the breeder in its own visualizing interface, which can be used then to make its selection. The high capacity of scripting offered by the data management systems of Dorian can address your specific breeding index designs. So let's you can share with us some examples. So today I would like to conclude my speech by the importance of now in plant breeding programs taken by analytical information. It results from certainly the strategy to imply into the development of varieties and hybrids which address new consumer and market needs. It results also certainly from a better understanding by the science of biological processes which can now be used in plant breeding programs. It implies to integrate into the breeding pipeline further activities with new biological traits to characterize and track, with the same constraints of multiple years processes and with the same high impacts of environment on the cultivar responses. Notably, it has been characterized those soluble solids of strawberries, which impact sugar content, is more dependent on environmental conditions than genetic differences among cultivars. So in this context, setting up an integrated tool offers the opportunities to make the data workflow smoother with less data exchanges, and that should save time and reduce the risks of errors. And finally, I think new technologies offer great opportunities to optimize the use of analytical information, and particularly sensory information, which are so important for the consumers, notably by confronting this analytical information with phenotype and genetic information. Some initiatives emerged on molecular markers developments associated with quality and sensory traits that are valued by targeted consumer and markets, and it should be a fantastic tool then for selection of promissory genotypes from early stages of the breeding process. Thank you very much for joining us, and let's see in another webinar. Thank you very much. Goodbye. Take care.
R&D experience, leading researchers to their goals.